Okay, so let's get started. You can see the screen, yeah?
Yes.
Okay, great. So this is the final lecture of the developmental biology class. This is meant to be a review lecture, but I've missed one, so let's go through this quickly. And so today I'm going to talk about the development of the head. And what I want to be able to describe is how we can see different faces or see different expressions, and how those, what the anatomy behind these expressions, how that's laid down, and how that develops. And so when we think about the tissue types in the embryonic head, as a kind of review, can you tell me what tissues we should be thinking about when we're thinking about this part of our body? In the embryonic head, what's there? Anyone?
The brain.
Yep, that's one.
Then you have muscles and blood vessels and skin.
Yeah, exactly. Yeah. So all of these. So you have absolutely, Shubha. You have muscles, blood vessels. You have skin. Let's include the other components of the skin, things like sweat glands and sebaceous glands. You have other kinds of glands within the head as well. You have the vasculature, both blood and lymphatic vasculature. You have the jaws as well. You have sensory organs. You have hair. You have, as well as the brain, the central nervous system, you have peripheral nervous system. And so all of these are laid down in the embryo from these particular types of tissues. And so you have neural ectoderm, which is going to give rise to the brain. You have mesoderm, of course. So mesoderm will be there. Mesoderm will be split into precaudal mesoderm and paraxial mesoderm. You'll have endoderm as well. And you'll have these components here, neural crest and cranial placodes, which we'll talk about in a little bit more detail in this lecture. Okay? Okay. So when Bhavana and Sonia gave their talk, and I talked about this as well. We've talked about the formation of the neural tube, and the neural tube is the precursor for the CNS, and all of you know that as the neural tube forms, we have this border region here, just between the surface ectoderm, and so the surface ectoderm will give rise to the skin generally, and the neural ectoderm, which will give rise to the CNS. And there's a border here, which is something special. And so this border will give rise to a structure called neural crest and to cranial placodes. So what we're going to do today is talk primarily about neural crest and cranial placodes and how they form, how they give rise to various components, and how these would be integrated to form, for example, cranial nerves, how the neural crest will give rise to jaws, for example. And then and then the open question of how muscles form as well. What we won't talk about is how the blood vasculature and the lymphatic vasculature forms, how glands such as salivary glands form, but I'll give you the basics so that you at least are aware that neural crest exists and that cranial placodes exist as well. And at any point, if you have questions, please interrupt me. I know it's early, but let's see if we can just interrupt me if there's anything unclear. So as I mentioned, the neural crest forms from that border between the neural plate and the surface ectoderm. In here, you're looking at this as the greens, this green strip here. We'll talk a little bit more about this in a few slides. So this border actually has two properties. So it will give rise to both the neural crest and it will give rise to sensory cranial placodes as well. The part of the border that's closest to the neural ectoderm will give rise to neural crest. So neural crest is specified as the neural tube is forming. As the neural tube closes, a specialized group of cells within these neural crest precursors delaminate and leave the neural tube. And they'll migrate away. And they have a unique property to be able to form a multitude of different types of cells. And so neural crest will form cartilage, bone. It will form sensory ganglia. It will form tooth precursors, pigment glands, and so on. And so all of these cell types that you're seeing here listed under either cranial to sacral neural crest come from individual neural crest cells that are able to form a kind of stem cell population that is able to give rise to these particular structures. Now, what neural crest is able to give rise to depends very much on its the position from where it's come from. So neural crest that's in the trunk, for example, can give rise to, for example, dorsal root ganglia, and cranial neural crest can give rise to chondrocytes and osteocytes. But but trunk neural crest will not be able to give rise to chondrocytes and osteocytes. So these are so even though the neural crest is a stem cell population and is able to give rise to different cell types, the potential of these stem cells is limited by the position from where it's arisen. And I'll show that in the next few slides. So do we have any questions up until now? Okay. The other cell type is that's coming up is the cranial placodes, and we'll talk a little bit more about the cranial placodes in a little bit. But essentially, these will form some of the sensory organs of the head, and this also includes actually the adenohypophysis of the pituitary. So part of the pituitary gland also comes from outside of the neural tube, and that's a cranial placode. The olfactory epithelium, so the olfactory sensory structures will come from the outside of the neural tube. These structures here, which we'll talk about in a little bit, the inner ear, all come from cranial placodes. And so we'll talk about this in more detail later. But let's shift back to neural crest. So neural crest is forming from this border between the neural ectoderm and the surface ectoderm. And a lot of work, primarily from the lab of Marianne Bonner, has gone into finding the mechanisms of how this neural crest has arisen. And so it's been thought that this border between the surface ectoderm and the neural ectoderm is important for the specification of the neural crest. And indeed, that's been shown to some extent. So there are signals that are coming from the surface ectoderm that are interacting with signals that are within the neural ectoderm that set up the pre-migratory neural crest, so the early neural crest. These set up these kind of genetic networks, and so the gene networks that establish the neural crest have been established. I don't want to go into them in too much detail. Suffice it to say that it's very well established, and so these kind of circuit diagrams that show which transcription factors turn on which transcription factors within the neural crest have been well established. In the three main ones for our consideration are these here. This transcription factor called FOXD3, this other one called SOX10, and ETS1 are all important for setting up neural crest. Knockouts of any of these affect not the whole of neural crest development, but some aspects of neural crest development. These genetic regulatory networks have been further subdivided into thinking about the types of cells neural crest is able to give rise to. And so here it's starting to think of neural crest as I mentioned, a stem cell population. And then if we're thinking about stem cell populations, how then different types of potentials are realized within the neural crest. So very similar to the way we think about development, neural crest development also is kind of hierarchical. And so you establish kind of broad modules that specify the neural crest, and then individual pathways after that that set up individual differentiation pathways. And so, for example, here, this specification module sets up a ground state which is acted upon by different types and different combinations of genes, which will set up, for example, neurons and glia, which will set up chondrocytes and osteoblasts, which will set up melanocytes. And these kind of studies have very, very well established this stem cell nature of neural crest, which is something we'll talk about in the context of the plasticity of neural crest, which is important when we're thinking about kind of some of the properties of neural crest that allow it to that allow different kinds of phenotypes to be explored. And so I don't want to talk too much about this, but those who are interested are directed to several reviews from Marianne Bronner, which are uniformly excellent. She's a great writer, and she has done a lot of work in understanding the regulation of neural crest. Okay. So I think we have talked about this, that one of the nicest things about neural crest, or one of the clearest properties of neural crest is that it forms by epithelial mesenchymal transition. I think all of you remember epithelial, this EMT process. Does anyone need a quick overview? A quick reminder. Good. So EMT, as its name suggests, is epithelial to mesenchymal transition. So remember, the neural tube is an epithelial cell type, and neural crest is mesenchymal. And so the neural crest is born within the neural tube in this border region, and it has the unique property of being able to switch its cell phenotype from this epithelial cell. Epithelial cells, remember, have junctions. They attach to the basal lamina, and to a mesenchymal cell type, which doesn't have junctions, which is free from the basal lamina, which is able to migrate. And again, the mechanisms in which neural crest is able to leave the neural tube are very well worked out. And I'm showing here this video from Paul Kaluza and Scott Fraser, showing the migration of neural crest. And so let's play this again. And so what you're looking at is the hindbrain of a chick embryo. And you can see here bits of the hindbrain. What you're looking at is R3, R4, and R5. The neural crest has been stained in so that it's fluorescent. And this movie will show you the neural crest being born in rhombomer 3 and 5, and then migrating out into various streams. And so let's play this again. And so you can see neural crest is actually born throughout the neural tube, but then it's forming these distinct streams here. So neural crest is leaving by EMT, by epithelial mesenchymal transition, but is exiting the neural tube using specific routes. And you can see these streams here and here. And we'll talk about these streams because they represent a fundamental organizational unit of the head, particularly of the face, that comes into play here. So these are branchial arches, and so what these crest streams that you're seeing here are leading to are branchial arches, and so you can see them leaving the neural tube here, and they will so here, and they will enter these bulges on the side of the head, which are called branchial arches. So what you're looking at here is a human embryo at 32 days. You're able to see the rudiment of the eye here, the rudiment of the nose, the nasal placode, and these here are the branchial arches. And so there's one that will form what's called the maxillary arch, the mandibular arch, and a few other arches as well. So branchial arches are a very conserved feature of vertebrate development. The branchial arches are called alternatively pharyngeal arches as well, and which gives us the phrase pharyngular as well. So the stage at which you can see pharyngeal arches or branchial arches is also called the pharyngular stage of the embryo. Branchial arches are these outpocketings on the side of the head. What you're looking at here is a sketch of a fish embryo, taking a section through the top of the branchial arch, or the pharyngeal arch it's called here. And when you look at this, what you can see, these bulges here are bulges of neural crest on the outside, mesoderm in the core, and there's a blood vessel running through the center of the branchial arch as well. It's surrounded on the outside by ectoderm, on the inside by endoderm. The endoderm here is in green. And at particular points, the ectoderm and endoderm meet, so they have no intervening mesoderm. And at these points, what we have is what's called the pharyngeal pouch, or branchial pouch, or pharyngeal cleft. Okay, and so these are just where the ectoderm and endoderm have meet or have come very close together. And so you have a series of these running along the side of the embryo, and in fact, in some fish species, you can have as many as 10 or 11 branchial arches. In us, we have 4 or 5 that are prominent, and these will give rise to fundamental units of the face. And so the branchial arches in us have been modified that go on to form, for example, the maxillary arch here will give rise to the upper jaw components. The mandibular arch will give rise to lower jaw components. Arch 2 is a hyoid arch, which will give rise to some of the support structures within the throat. And so all of these are kind of modifications of a particular plan. This fundamental repeating unit, the branchial arch, can be modified by different genetic programs to give you slightly different phenotypes, and I'll show how that one mechanism in which that's possible in the next couple of slides. So in fish, some of these components, as I mentioned, in fish is no different. Some of these arches are modified for particular phenotypes, and so here the second arch, the hyoid arch, gets modified to form the orbicular flap, which generally covers the gill slits. And so this is an important thing to note, because these points at which the endoderm and ectoderm meet, these clefts or pouches, do actually form a particular purpose in fish, for example, and lower vertebrates. And so these will become gill slits and allow the flow of water through the respiratory epithelium. And so, and in higher vertebrates that have moved onto land, these gill slits, of course, are no longer needed. And these have been modified for different purposes. And I'll let you understand those, perhaps by looking at this review here, okay? So any questions, particularly about gill slits, if you have any? Great. So as I mentioned, the branchial arches are important organizational units for neural crest. So the neural crest does leave the neural tube, and it flows in particular streams. And so crest, for example, that leaves the forebrain region will flow into what's called the frontonasal process. And so this is this is essentially this part of the head. So it will form some of the upper jaw, some of the neural crest and mesenchyme. That's just in the underneath the forebrain. It will also form some of the structures within the maxillary arch as well. So things that will form the upper jaw, and so on. This kind of restriction of neural crest streams is also very important. And this restriction is mediated by classic gene proteins that are involved in repulsion and attraction in neuronal systems as well. So things like the semaphorins, the neuropilins, the ephrins and the epinephrine system are all involved in neural crest migration, and they're all their function is repeated when we start thinking about patterning of neurons as well. And so perhaps Bhavna and Sonia spoke about neuronal patterning and the formation of neural circuits, which do involve things like semaphorins and neuropilins and epinephrines in those systems as well. And so this kind of brings another point. These kind of molecular systems are involved in patterning and segregation of the neural crest, or in other systems such as when we're thinking about the convergence and extension migration during gastrulation. These molecular systems are used again and again. And so this molecular toolbox is being used in different contexts to do kind of the same thing. So semaphorins will be used in the patterning of the in retinal tectile projection. So the pathfinding of axons from the retina, for example, to the tectum, also use the semaphorin, neuropilin, epinephrine system as well. And so that's that kind of brings up an important point, that if you're trying to understand your particular system, and you may be working on neuronal patterning, you may be working on pathfinding of vasculature, for example, it's always worth looking at different model systems, for example, neural crest, to inform you about what may be happening in vasculogenesis, for example. As a neural crest migrates, for example, it migrates with a direction, and this direction seems to involve also then the planar cell polarity components, Rho-RAC, for example, which we've spoken about before in the context of gastrulation. Okay. So once the neural crest gets to its destination within the branchial arches, and you're seeing this from this figure from this review both from Filippo Ridgely, it starts to differentiate into jaws. So the jaw components. So it will differentiate, of course, into other components as well. But for this lecture, I want to talk specifically about the jaws. So what you're looking at here is kind of the lineage maps of three different vertebrates. This is a chicken skull. This is a mouse skull. This here is a human skull. And what you can see is where the bones of the skull have come from. And so what you're looking at here in this slightly pixelated image, if you can't see it so clearly, I'll explain it to you. You're seeing the different populations of neural crest that have come from different parts of the embryo. And so remember, the neural crest is coming from the neural tube. And so you have neural crest that's coming from the mesencephalon and diencephalon, so from the forebrain and midbrain. And these will end up in the frontal nasal process, and some in the maxillary arch. You're having neural crest that's born in different components of the hindbrain as well. And so I think Bhavna would have spoken about rhombomeres to you. Is that correct? Hello? Sorry, can you hear me?
Yeah, Raj.
Okay.
I don't think she's talked about rhomboids.
Rhombomeres, yeah? No?
Yeah. No.
Okay, so rhombomeres are a similar organizational subunit in the hindbrain. They're bulges in the hindbrain that give rise to various components. They're segments within the hindbrain, and you can look them up if they haven't been if you haven't been introduced to them. They're not they're kind of important patterning components, and so they're used primarily during early development to organize the development of the hindbrain, but they also are sites where the neural crest is born. And so, for example, here you're seeing R1, R2, R3 are rhombomeres 1, 2, and 3. They'll form neural crest, which is heading towards branchial arch 1. These rhombomeres here will form a crest that's heading to branchial arch 2. This rhombomere to branchial arch 3. And so that's the only thing that's important there. Okay, so so you have, of course, then the skull. The skull has bones that are formed from these various regions. So here is a frontal bone, for example, which is this bone here. This is primarily derived from the frontal nasal process. You have things like the maxillary and zygomatic bones, which are formed from neural crest that comes from here. The dentary, which is coming from branchial arch 1. Hyoid, the support structures for the respiratory system are coming from different populations of neural crest. So this is kind of this is important because then what you're thinking about is how these different populations of neural crest can be patterned and regulated. So it's known that so some classic work. So how do we know where this neural crest is coming from, and how do we know that it's patterned? So here we are having here we have classic transplantation experiments from Nicole Le-Doran's group, and she's been looking at this for over 50, 60 years, and what she's been doing is transplanting. Do you have a question? Okay, so what she's been doing is transplanting bits of the brain from a quail embryo into a chick donor, and asking where the crest goes to. And so here, for example, she's been taking kind of crest that's from the midbrain and forebrain that should give rise to branchial arch 1, and then transplanting it further down. And so removing this part of the embryo and replacing it with this crest here. So we're having two streams of crest that are from that are forming branchial arch 1. And what she's found is that these muscles, so these jaw joints, are still laid down. And so what you're seeing is now a chick embryo that has two mandibular bone pieces, and so a second lower jaw here. In some cases, she's able to show that some of these neural crest precursors have plasticity, and so they can be moved to different regions, and they don't form this these second arches, and so they will, for example, you can take a more caudal piece and move it further anteriorly here. So rhombomer 4 that should form part of the hyoid structure, you can move anteriorly, and that will be incorporated very nicely within the existing mandibular arch. And so there's a certain amount of pre-patterning, there's a certain amount of plasticity that exists within the jaw, and it depends on which exact component we're thinking about. So what does pattern the jaw then? And so how do these kind of repeating segments know that they're going to become the lower jaw, for example, or the upper jaw, or the hyoid support apparatus? And it seems that, and this is now classic embryological experiments from Nicole Le-Doran, and it seems that actually the endoderm is important for imparting that information. And so the endoderm, remember, is underneath all of these structures. It's forming these cores within the branchial arches. And what has been done is to take various strips of endoderm and transplant them into ectopic locations. So this is from a quail. It's going into the chick, And if you remember, the superpower of quail cells is that they have an antibody that recognizes them, so you can know you know where your transplant has gone. And what you can see here is this endoderm is able to induce a new cartilage element. And when you let these embryos grow, what you can see now is a chick. And so what you're looking at is the result of this transplantation. You have an upper beak, you have a lower beak, and now you have a second lower beak as well. And so this MC here is the Meckel's cartilage, which is part of the lower jaw. And you can see now you have a duplicated Meckel's cartilage. And so this is exciting and interesting. It tells you that there's crosstalk that's happening between all of these tissues, between the endoderm, neural crest, and remember the neural crest has a pre-pattern as well from has some extent of pre-pattern as well. And all of these kind of different conversations are happening within the face to give you particular jaw structures. More than that, so we're talking a little bit about the plasticity of neural crest, but now let's talk about where it's not plastic. And so this is work from Richard Schneider. And what he's been able to show is kind of the species-specific program of neural crest. And neural crest has autonomous information that is able to drive particular species specificity. And what do we mean about that? And so the classic experiment that he did was to take a quail neural crest and transplant them into duck. And if you look at the difference between a quail and a duck, you can see that their beaks are very, very different. That quails have this kind of pointed beak, ducks have a bill that goes like this. And if you then transplant the quail donor into a duck host, what you now see is on the donor side, the beak having a quail morphology. And so remember, this is a duck host now with a quail-like beak. Okay, and so there's some autonomous program within the neural crest that provides shape. Rich also showed later that when we're looking at things like the formation of cartilage, that cartilage formation, the kind of timing of when cartilage will chondrify depends on the donor tissue. There's no non-cell autonomous factors that set up the chondrification process. This is all set up by within the neural crest itself. Okay. So this is this kind of experiment is incredibly important. I think it tells us that the neural crest has plasticity, of course. It has its own program as well that is set up much earlier. And so what's not clear is when if you were to take a quail donor and put it in a duck host, is there a stage at which the quail donor would be able to take on a duck-like morphology? And then what's driving that duck-like morphology, this kind of action of the this bill morphology as opposed to this beak morphology? And so that's still an open question. Ways to think about it, of course, we can think about this in terms of Darwin's finches as well. And this may not be such an easy, it may not be such a clear thing to think about. And so remember, I'm sure you've all read Origin of Species. You know that some of Darwin's thoughts on variation and natural selection have come from looking at the different types of finches in the Galapagos. The finches there have lots of different types of, there's a number of different finch species with different beak morphologies. And they're all essentially related. And depending on kind of what they're feeding on, they have different beak structures. And this was found to depend on a single gene, calmodulin, and how it's interacting with the neural crest. And so different levels of expression of calmodulin or different ways in which it's deployed seem to dictate the morphology of the beak. And this beak morphology could only be driven because the neural crest itself is very plastic. And so remember, I told you it was a stem cell population. And because of its very nature, because it is a stem cell population, it can be acted upon by non-cell autonomous signals to change the morphology a little bit. And so we're getting to a point in which we're understanding that neural crest has these non-cell autonomous factors which drive particular processes like the timing of chondrification, some aspect of shape. But you also have these outside signals coming from the ectoderm or from the endoderm, which are able to drive morphology as well. The exact kind of mix of both and how that drives the shape of the face is still really unclear. And hopefully within the next 10 or 20 or 30 years, we can start understanding how these kind of conversations between different tissue structures kind of drive facial patterning. So as well as Darwin's finches, you have bats as well. Bats, of course, are a mammalian species. They also have different snout morphologies depending on their diet. You have some bats that have a long face, and these are nectar feeding bats. Other bats that have a truncated face that eat fruit. And so this also seems to depend, similarly to the finches, it depends on signals that are coming from the facial primordia that are dictating how the neural crest behaves. And so this is from this very nice review from Arkat Abramova from 2017. He's shown very nicely that some of the same mechanisms that are happening in the finch seem to be also happening in the bat as well. And then one thing I've always been interested in are dogs. And so remember, dogs they're a single species, and you have so many different types of facial morphologies, so many different types of morphologies themselves that seem to tell you that this kind of neural crest plasticity is important in setting up these different phenotypes of the face. And how this is controlled will be very interesting to find out. So as well as jaws, we have muscles as well. Muscles are coming from the mesoderm. So whereas the jaw elements come from neural crest, so the neural crest derived mesenchyme, muscles come from, on the whole, from mesodermally derived mesenchyme. One thing that's not clear and is really an unknown is how then jaw elements and muscle elements kind of co-pattern. And so it's likely that jaw elements dictate the pattern of head muscles. So when we're thinking about, for example, the mandibular muscles or the jaw adductors, for example, how these are set up and how they insert into different jaw elements is still very, very unclear. and these are open questions in the field, and that's and I've left it at that for the jaw muscles, because so little is known about how the jaw muscles are forming. I'll end this on now on you talking a little bit about cranial nerves. And so I think we all know what the cranial nerves are. Can anyone tell me how many cranial nerves there are in the human head?
You have 12 pairs.
Fantastic. Yes. So there are 12 pairs of cranial nerves. Thanks, Shubha. We go round olfactory, oculomotor, the trochlear, abducent, trigeminal, vestibular cochlear, glossopharyngeal, and so on. Okay, and so these are all important nerves that drive different components of facial function. So either motor or sensory function in the head and the face. We'll talk about the sensory ones from now, but I will tell you a little bit about the embryology behind some of the cranial nerves. Again, the cranial nerve organization seems to follow very similarly this idea of branchial arches. Each arch has a particular cranial nerve that goes through it. And each arch, each cranial nerve is derived from a specific population of a specific progenitor population. And so what I want to talk about is these nerves here, the trigeminal ganglion, the geniculate ganglion, the nodos, and the superior and petrosal ganglion. And so these purple nerves here, which will give rise to the 4th, 7th, 9th, as well as the 8th nerve here, okay? And the olfactory placode, because all of these are forming from, and the 3rd, all of these are forming from cranial placodes. And so cranial placodes are the other side. And so if you remember, we have the neural plate border. On one side, on the neural ectoderm side, are the neural crest. On the surface ectoderm side, we have what are called cranial placodes. And so these will give rise to particular sense organs. They'll give rise to the olfactory system. There's a cranial placode that will give rise to the lens. Because the lens doesn't have any neurons, we can skip that for the time being. It'll give rise to the trigeminal nerve as well. So both ophthalmic and maxomandibular branches of the trigeminal. It'll give rise to the inner ear, the otic. So this is the otic placode, and it'll give rise to these 3 nerves here, the geniculate, petrosal, and nodos as well. And these olfactory, trigeminal, epibranchial, otic all form major cranial nerves, and we'll talk a little bit about how they form now. So in the same way that the neural crest has a particular developmental program that's been well worked out, and it seems to involve a hierarchical segregation or hierarchical organization of its development. Cranial placodes are the same. And so remember I told you that they form from the border region between the neural ectoderm and the surface ectoderm. They form in what's called a preplacodal domain. And so this whole strip of ectoderm that surrounds the neural plate, which will form the CNS, seems to be competent to give rise to cranial placodes. And this is marked by these 2 genes here, 6, 1 or 4, and IA1 and 2. And this strip seems to be very important for the subsequent formation of these cranial placodes. And so what you can see here is this strip of orange tissue giving rise to then these particular placodes here, otic, epibranchial, trigeminal, the lens, olfactory placode, and adenohypophysis, the pituitary placode as well. Now, there's some controversy on whether this strip, this orange strip, the preplacodal domain, is one preplacodal domain or two preplacodal domains. So some people, and I'll show my bias here because it's me, I'm one of those people, like to suggest that there's two preplacodal domains, an anterior and a caudal one, and others will suggest that there's a single preplacodal domain. This is kind of a detail which may or may not be important, but when we start thinking about where in evolution these kind of particular structures could have arisen, and the relationship between the olfactory placode, for example, and the otic placode, they become, I think, they cross this kind of semantic border, I think, and so it's something to keep in mind. I'm going to, just because it's my research, I'm going to quickly talk about what's known about how the inner ear forms, just as a way of describing the induction of a placode. And these kind of hierarchical steps that the preplacodal domain takes to subdivide into a particular sensory placode. So the ear, as you all know, is where hearing and balance take place. And this kind of fairly complicated structure that you're seeing, this is the mouse inner ear, comes from just a single placode. And this placode is set up early, and it's set up on the surface ectoderm here. And so what you're looking at is a chick embryo at different stages. What seems to happen in the formation of the otic placode is again this subdivision of potential. So I told you that this strip all around the neural tube, and by now the neural tube is closed, is able to give rise to sensory placodes. By a particular stage, we seem to have subdivided this preplacodal domain into what is now the precursor domain of the otic placode and epibranchial placodes. So I've called it, it's either called the otic epibranchial precursor domain, the OEPD, or the posterior placodal area. This structure is then further subdivided into otic placodes, so the inner ear precursor and the epibranchial placodes, and then the epibranchial placodes themselves are further subdivided into the individual geniculate, petrosal, and nodose ganglia. So this kind of subdivision is all well and good. We can go further and understand what the signaling mechanisms are. Again, similar to what we discussed for neural crest. In the inner ear, we also have this kind of crosstalk between between signals coming from different parts of the embryo acting on other parts of the embryo. So we have signals initially coming from the endoderm, which pattern the mesoderm. We then have mesodermal signals which pattern the surface ectoderm and the neural ectoderm, which then work together to then set up the posterior placodal area. and so on. And so you have this kind of continuous conversation between different tissues to drive development and induction of particular structures. And so using this kind of this fairly elaborate crosstalk of signals, we're able to start getting the otic placode. And this then forms from the surface, and it will invaginate and then form an inner ear structure, which we all know about. So some of these placodes also make neurons. Almost all of them do. The otic placode or the inner ear will make its own innervation system. So the cochlear vestibular ganglion is made by the inner ear. The geniculate petrosal and nodos also form neurons. So the geniculate forms neurons that will innervate some of the tongue, for example, sensory neurons that drive, that will innervate some of the ear, some of the actual pinna of the ear, and so on. And so these, even though they're born outside of the nervous system, they need to find their way back into the nervous system to make a cranial ganglion. And this seems to be driven again by a crosstalk between neural crest and placodes. And so remember, we have this beautiful organization of cranial nerves. They seem to be segmentally organized. They seem to sit at the top of these branchial arches, and then they feed back into particular structures within the brain. And I'm going to end on a conversation that happens probably between these placodal derived neurons and the neural crest, which seem to drive this final stage of cranial nerve formation. And so this is beautiful work that was done from Jo Begbie some 7 years ago. And what she found was that almost all of the cranial placodes, because this is certainly true for the otic vesicle as well, use the crest streams that are laid down when the neural crest migrates out. The placodal neurons can, as they form, will use these crest streams to migrate back into the brain. And how is this happening? It seems that the neural crest encapsulates these placodal derived neurons. And so what you're looking at here, for example, is the chick embryo. This is stained with a neural crest marker and neurofilament, as well as DAPI. And what you can see is the neural crest is wrapping around. Here, islet1 is a marker for the placodally derived neurons, and you can see the neural crest is encapsulating the placodally derived neurons. So it's forming a sleeve, a tunnel through which the placodally derived neurons can travel back into the brain. And so this is shown here quite nicely. This is the geniculate placode here. This is the geniculate neurons. They're being born outside of the nervous system. You can see here this crest stream from rhombomer 4. And when we look to see where these neurons are, you can see that they're traveling along this crest stream into the hindbrain. And so in this way, you're setting up the segmental arrangement of cranial ganglion. And again, just from an interaction between neural crests and cranial placodes that help the cranial nerves back and find their way to the CNS. And so that's, in a nutshell, some features of head development I wanted to talk about. I think that's it. So if you have any questions about what I've talked about, then you can ask them. Okay. If you don't, then if you have any questions about any other aspect of the course, then you can also ask them now. Okay. All right. So it's great. If you don't have any questions, if there's nothing you want to discuss, then well, thank you. Yeah. So thanks for being part of the course. I will now put up the end of term paper for you all. You have, I think the deadline is middle of February for this. So you have some time to think about what you want to write about and then write it. You can ask me if you want to kind of pre-discuss your ideas, then feel free to contact me. As many of you know, I'm sometimes quite responsive and sometimes I miss your email. So don't be afraid to write to me more than once if you haven't received a reply. So either me or Varsha. I think Varsha's here. Are you Varsha?
Yes, Raj, I'm here.
Yeah. Okay. We'll be happy to talk over some of your ideas. Okay. Cool. So, well then, if not, I'll let you all go. Have a lovely day. Have a wonderful new year, and see you next year. See you in person next year. Okay.
Thank you, Raj.
Okay.
Thank you.
Happy New Year.
Thank you,
Happy
Raj.
New Year.
See you, Raj.
Happy New
Thank
Year.
you. Happy New Year.
Happy New Year.
Raj, I had a clarification to make.
Hi, Sugar. Thanks.
Regarding the move, students, like some of us are coming from Bangalore, and
Yeah.
we were told that it's not necessary for us to quarantine. So what is the protocol for us?
So I was going to meet all of you individually, or in a group. Yeah. So I think so. It's you and Sahana's here as well, and Garish. Yeah, coming from Bangalore. So we'll you can come straight into the campus after everyone should have finished their quarantine, and so that will be about the 10th, I think, of January. During that time, there's no protocol. The security will have your details already. If you want to come into campus before then, if you want to start talking to PIs, you can. Okay. Just let me know, and I'll let security know so that they can prepare your so that they at least know you're here. Okay. But there will be an orientation from the 10th for a few days for all of the batch, just to show you how the campus works, take you through various features of the campus, and then what will happen is you will spend a week talking to the PIs that you might want to rotate with. And then start your rotations. Okay. So Shubha, what would you want to do? Hello, Shubha?
I wouldn't mind coming in on the 4th itself.
Okay. Okay. So then yeah. If so I'll send you all a message about that. Okay. And just send me a reply with what your plans are. Okay. Yeah.
Okay, Raj, thank you.
Yeah, and I'll contact you all today to set up individual meetings for you as well. Okay. Yeah.
Okay, Raj.
Raj,
Okay.
I also have something else to ask you.
Hi, Nivedita. Yeah.
Hi,
go.
yeah. So the thing is, so when we'll be coming to campus, we'll go straight into quarantine. So because our baggage is, the allowance for the baggage weight is limited,
Uh-huh.
is there any like person allotted who can like buy us essential stuff if we need it during that period? Because I don't think we can bring too much stuff, and neither can we go out and buy them ourselves.
Yeah. You can't go out and buy them.
Mm.
Yeah, we'll sort that out.
Okay.
I'm sure that I can ask some of the previous batch to help you out.
Yeah, we can just get in contact with students who can move around. We can do
Yeah.
that.
Yeah. So I'll talk to last year's batch, okay, and see if we can set up kind of some support mechanism for those of you in quarantine.
Okay.
Yeah. You also have, of course, Shubha and Sahana, who are yeah. So I mean, I think Shubha maybe also, both of them, Shubha and Sahana, maybe kind of uncertain where everything is, especially within the campus. So it may not be the best way, but certainly there's a way of getting things to the caretaker who'll be bringing you food, and then him bringing things to your room. Okay.
Okay. Thanks.
Okay. All right. Thanks, everyone.
Thanks, Raj. Bye.
Bye.
See you.
See you.